Okay, next is on DNA or RNA vaccines. So basically these DNA and RNA vaccines use part of the virus' own genes to stimulate an immune response. So basically the principle behind the DNA vaccine is injection of plasmid that contain the DNA that encode the protein antigen of the pathogen. While mRNA vaccine is a vaccine that consists of mRNA strand that codes for a disease-specific antigen. Once the mRNA strand in the vaccine is inside the body cells, the cells use the genetic information to produce the antigens. This mRNA strand is like conveying the message to produce the desired protein antigens of the pathogen. So, one of the best example of vaccine that utilize this concept, which is the mRNA-based vaccine, is the COVID-19 vaccine produced by Pfizer and BioNTech. So, basically, the vaccine delivers strands of genetic material known as mRNA, which turns people cells into the spike protein factories producing. Then the cell display the protein piece on its surface and stimulate the necessary immune response to confer protections against COVID-19. As we can see here, with a DNA vaccine, the virus genetic information is transmitted to other molecule that is called the messenger RNA. I try to find the... Okay, you see, for the DNA vaccines, it need to go into the nucleus, being read here, transcribes, transfer to the RNA, and the RNA will, in the form of the messenger RNA, send the information to produce the protein here. But with mRNA vaccines, actually it is one step ahead of DNA vaccines. So this is where the mRNA strands, which is straight away can convey the message to the cells to produce the particular antigens. So, in other words, mRNA vaccine is not being integrated into the genetic material, thus cannot alter the people's DNA as being claimed. And the mRNA will just be degraded once it sends the message. Next is on vaccine adjuvant. Basically, an adjuvant is an ingredient used in some vaccines to create a stronger immune response. In simple words, adjuvant help vaccines to work better. Some vaccines that are made from a weakened or killed microorganism contain naturally occurring adjuvants and help the body to produce a strong protective immune response. However, most of the vaccines or the modern vaccines being developed now include just a small components of the microorganisms, be it just a protein, DNA or RNA based vaccines, which is fully immunogenic on its own. Thus, this type of vaccines required administration with adjuvant to work effectively. So in this advanced era of technology, the search for new vaccines quite challenging, and sometimes you can't simply use the traditional approach where you can just weaken or inactivate it. Sometimes you need to have a more fancy approach which will generate a better safety profile, but you also need to search for an effective adjuvant to be used in concordance with these vaccines to generate the immune response that can confer protection. So basically, a key adjuvant function is to overcome the poor immunogenicity of subunit vaccines by improving pathogen recognition and eliciting a response similar to the innate immune response. And recently, nanoparticles technology are of great interest to be utilized as vaccine adjuvants. So nanoparticles can be made from various types of biomaterials, like from the virus-like particles, from the liposomes, where all these can form the nanoparticles, and the antigen can be entrapped inside these nanoparticles, thus provide protection to the vaccine antigens. It can be made from a polymer and also non-gradable nanoparticles. Why nanoparticles? Because nanoparticles resemble a similar size dimension to the virus or to the pathogen, so that it can be highly recognized by the body, by the immune system, so that it can be acquired and activate the necessary immune cells like the antigen presenting cell, so that our body system can recognize this and further translate the encapsulated antigens and to produce the necessary immune response against these particular antigens with the help of the nanoparticles based vaccine delivery platform. So next, we will have a look on the general procedure for vaccine development. So the general stage of the vaccine development cycle can be comprised of the exploratory stage, the preclinical evaluation, where mainly done on animals, and then the clinical development, where this involve the clinical trials, regulatory review and approval, and the next step is on manufacturing the vaccines, and of course we will have the QC, which is the quality control. So the exploratory stage involve a basic laboratory research, which is to identify natural or synthetic antigens that might help prevent or treat a disease. And these antigens could include virus-like particles, weakened virus, weakened bacterial toxins, or other substances derived from pathogens. And I myself, my research is mainly involved on vaccine development. Now we have few vaccine related research, which is on the dengue virus and also again as aureus. But interestingly, I also have one project on a therapeutic vaccines. Of course, all this while we always think that vaccine is to act as prophylactic, which is to prevent, but we can also utilize vaccine to act as therapeutic approach, which in here I developed a breast cancer vaccine, therapeutic vaccine. Alright. And then we will have a pre-clinical stage. The preclinical and the exploratory stage is somehow quite highly interconnected to each other. Basically, preclinical studies use tissue cultures or cell culture systems and animal testing to assess the safety of the candidate vaccines and its immunogenicity or ability to induce a significant immune response to confer protective effect. Animal subjects may include mice and monkey. For example, for dengue virus developments, we need to infect the monkey in order to test the vaccines, because monkey is the only animal that can develop the symptoms similar to human for the dengue virus. So basically, these studies give researchers an idea of the cellular response that they might expect in humans for the vaccine candidate that they have developed. In this stage, they may also suggest a safe starting dose for the next phase of research, and the vaccine will also be improved to make it more effective and safer. Like I mentioned earlier, in this stage, they may also do challenge study. Challenge study is where the animals, once they vaccinate, they will be infected with the targeted pathogen or the microorganism that the vaccine wish to confer protection against. In phase 3, it will involve the longer trials, like thousands to 10,000 of people. Phase 3 are randomized and double-blind and involve the experimental vaccines being tested against a placebo. And the goal here is to assess vaccine efficacy in a large group of people. And in phase 3, if the phase 3 is successful, a vaccine developer will submit a license application to FDA. Then FDA will review the result and the data. Once being approved, FDA will monitor the production of vaccines and review the manufacturer tests of lots of the vaccines for potency, safety, and purity. And then in the phase 4, which is somehow an optional study that the drug companies may conduct after a vaccine is released, the manufacturer may continue to test the vaccine for safety and efficacy throughout this phase 4. So basically, this infographic on how, on a general, vaccine is being manufactured, which take almost like 12 to 36 months to manufacture a vaccine. So you can see the pathway of how vaccine is being manufactured, from the raw material and the active ingredients and coupling and formulation with all the components of the vaccines, from the filling, and then you produce in bulk, which we call a lot release, and then including on its distribution. Standard vaccines require temperature between 2 to 8 degrees Celsius, but it's just recently that this mRNA based vaccine required ultra cold condition, which is -80. And then the distribution, the finished product is delivered to the distributors, and they all need to comply with this good distribution practice, GDP, on the cold chain and so on. And of course, next we have the quality control for the vaccine productions, which all the factory settings need to be complied with GMP, good manufacturing practice, and they are entitled to ensure that the end products are safe for use in humans, and there's always a checklist or monitor of the end products to ensure that the identity, strength, quality, and purity of the products are consistent. So these are some of the tests to study this consistency, like the identity test, which they can use PCR, serological test, or ELISA to measure on the presence of the protein, if it's protein-based antigen, and HPLC, analytical and preparative, to see the purity of the antigens as well. So identity tests is included to ensure the vaccines contain the exact antigen for the particular disease. And then they have the potency test, which is to demonstrate that each antigen in the products meets the recommended guideline level in internationally recognized tests. Means that the amount, content, the type of the antigen is sufficient to produce the necessary immune response. So we can done this by performing the bioactivity assay or specific binding assay. And then we do have a general test that looking at the appearance, the pH, osmolality, and by using the ion chromatography, and then also to evaluate on the toxicity, whether you have any trace of toxic compound, any residual toxicity. So basically the general test and safety test is to demonstrate the product contain no other harm material and they are sterile before being distributed. So basically this graphic summarizes some of key components in vaccines, as well as clarifying their purpose and safety in the concentration present. You may have a read and look this graphic in details on the description of each of the component, and it also provide some answer on what the anti-vax always claim on the side effect of a vaccine, and how these ingredients that are used in a concentration that will not cause any unwanted side effects. And this is the vaccine component of the Pfizer BioNTech COVID-19 vaccines, where the active ingredient is the mRNA strand that encode the S protein of COVID-19, and the lipids component here is the solid lipid nanoparticles that's used to produce the solid lipid nanoparticles, which act as the adjuvant to boost the immune response of the encapsulated mRNA antigen, while also to protect it from being degraded. So various types of salts are used that act as stabilizer, and we have a sucrose, which is a type of sugar which act as cryoprotectant. Why? Since the mRNA need to be stored in ultra cold conditions, we need the cryoprotectants to prevent damage to the cells during freezing.